In Part A of Chapter 8, we started looking at the steps to disease. We looked at how they gain entry through portals, so we looked at the portal of entries. We saw adherence, and we saw invasion. Now let's cover damage. There are 4 methods of damage. 1 is using the host nutrients. 2, direct damage around where invasion occurred. 3, toxins. Toxins are poisonous substances that are produced by pathogens. They are substances that contribute to pathogenicity, so a primary factor in pathogenesis. Toxicity is the ability to produce a toxin. What was toxemia? Toxemia was the presence of toxin in the host's blood. 4th method is hypersensitivity, and this is when the immune system overreacts, and we'll be covering this in a later chapter. The first way of damage is host nutrients. Iron. Iron is typically not available to pathogens. 2 reasons. 1, hemoglobin has a heme unit that has an iron component, and that allows it to bind to oxygen. Transferrin takes recycled iron and transfers it to the red bone marrow where we can make new red blood cells. A pathogen may secrete cytophores to use the host's iron. Cytophores take the iron from the host iron-binding proteins. The pathogen may secrete toxins to help to get iron. So it secretes toxins to kill the host cells to free up the iron. Second method is direct damage. Metabolism and production of waste in the host. So it produces waste products. Multiplication, and it disrupts the host cell function. Third is toxins. Let's start with endotoxins. Endotoxins are mostly gram-negative bacteria. They're a part of the outer portion of the cell wall in the outer membrane. They are lipids mostly. So lipid A is an example of an endotoxin. There's no fever. There is a fever associated with this, so yes, there is a fever associated with it. It's neutralized by antitoxin. When an antitoxin is antibodies against a specific toxin, so no, there is no antibodies against an endotoxin. The LD50 is relatively large. Most endotoxins exert effects when gram-negative microbe lyses. So you'll see here that we have an intact outer membrane, but then when this cell lyses, it releases the endotoxin. Exotoxins are mostly from gram-positive bacteria. They're produced inside of the cells and released to the outside either by secretion or lysis. So you'll see here that it's made on the inside of the cell and then released to the outside. Exotoxins are mostly enzymes, so small amounts can be quite harmful since they are enzymes. We know that enzymes can be used over and over again. Exotoxins are soluble in the body's fluids, so they easily dissolve into the blood and can be transported throughout the body. They are highly specific, so they are specific for a structure or function in the host cell. There's usually no fever associated with it, and yes, it is neutralized by an antitoxin, so the host can produce antitoxins. The LD50 is quite small. Vaccines can be made against them, and that vaccine is called a toxoid, and a toxoid is an inactivated toxin used in a vaccine. There are 3 categories of exotoxins: AB toxins, membrane disrupting toxins, and superantigens. AB toxins. The part A is the active enzyme component. Part B is the binding component. Here are the steps. The toxin is released. Part B binds to the host receptor and is transported to the cytoplasm. Part A inhibits protein synthesis of the host, and the host cell dies, and B is released from the cell. Example of this is the diphtheria toxin, Corynebacterium diphtheria. Let's take a closer look at the AB toxin. So remember, it's made on the inside, and it's a protein. So we're going to go through transcription to make the mRNA. Then we're going to go through translation to make the actual protein. Then it's released from the cell, right? As the toxin is released, the B portion binds, and then the A portion moves into, the A and B moves in, and then it inhibits protein synthesis. Membrane disrupting toxins. Membrane disrupting toxins disrupt the host plasma membrane. So they lyse the host cells by making protein channels in the plasma membrane and disrupts the phospholipid bilayer. There are two varieties. Leukocidins target phagocytes by putting protein channels. So example of this would be Staphylococci leukocidins. Hemolysins target red blood cells. They also produce protein channels. Example, Streptococci, which uses streptolysin. Superantigens invoke a very strong immune response. So they cause an intense immune response due to the release of cytokines from the host cells. So release a lot of cytokines, which can cause severe symptoms. So your T cells do that. They release a lot of cytokines. And symptoms would be fever, nausea, vomiting, diarrhea, shock, and death. So we see this with Staphylococci and food poisoning toxins. Another way to classify exotoxins: cardiotoxins attack heart cells, neurotoxins attack nerve cells, enterotoxins attack the lining of the GI tract, hepatotoxin attacks the liver cells, and leukotoxins attack the leukocytes. Some more important exotoxin examples: botulism toxin, which causes Clostridium botulism, is an AB neurotoxin, and it prevents the transmission of signal from the nerve cell to the muscle cell at the NMJ, also known as the neuromuscular junction, and this inhibits the release of acetylcholine. And if you remember from 1151, if you inhibit that, then there's no muscle contraction. Tetanus toxin, caused by Clostridium tetani, which causes tetanus, is also an AB neurotoxin, but this one goes to the central nervous system and prevents the inhibition of random contractions by the muscles. The cholera toxin, Vibrio cholera, also known as cholera, is an AB enterotoxin, and basically A induces cyclic AMP formation from ATP. And this causes the discharge of many fluids and electrolytes from the intestine. And this particular toxin is very heat sensitive. Endotoxins, we see problems with the antibiotic treatment. If a doctor prescribes you the wrong antibiotic, sometimes it could end up lysing the cell. And guess what? You go from feeling bad to what? Worse, because we know the endotoxin basically is released when you lyse that cell. And this causes macrophages to release high levels of cytokines. So, general effects of this would be 1. Disseminated intravascular clotting, in which the endotoxins activate blood clotting proteins which block your capillaries. 2. Septic shock. You can have a loss of blood pressure due to bacteria, endotoxic shock. Macrophages release TNF, which is tumor necrosis factor, when they attack some gram-negative cells. TNF binds to many tissues of the host and may cause damage to your blood capillaries, so loss of water and decrease in blood pressure, and then also damage to your blood-brain barrier, leading to infection of the central nervous system. And then the third effect would be what we call the extreme pyrogenic response. In extreme pyrogenic response, a macrophage has ingested an endotoxin or bacteria. Okay? And when the macrophage engulfs it, of course it's going to break it down, and that's going to lead to the release of the endotoxin from the bacteria. This causes the macrophage to release interleukin-1. Interleukin-1 is a pyrogen, and that pyrogen is then going to get into the bloodstream, as we see here, and it's going to travel to the hypothalamus. Once it gets to the hypothalamus, then that's going to cause the hypothalamus to release prostaglandins. And what the prostaglandins are going to do is going to reset the body's thermostat. And when you reset the body's thermostat, guess what you form? A fever. So how do we test for endotoxins to avoid those serious problems? Well, we can do what they call the limulus amoebocyte lysate assay, LAL. Horseshoe crabs. Does everybody know what a horseshoe crab looks like? Here's what it looks like. A horseshoe crab has amoebocytes, and these amoebocytes have lysates. And those lysates can cause clotting in the presence of endotoxins. So, if a patient has an endotoxin, we could do this test to determine if they do have this gram-negative bacteria, and if they do, we might want to prescribe another type of antibiotic that does not lyse the cell wall. Viruses can penetrate and grow inside of cells where the host defense can't get them. So then they have what we call cytopathic effects. Cytopathic effects are visible results of viral infection. So we'll see cell death by multiplying viruses, inhibition of DNA, RNA, or protein synthesis, and effects on the permeability of the membrane. Cytocidal versus non-cytocidal. Cytocidal effects results in cell death. Non-cytocidal effects causes cell damage but no cell death. So we can have macromolecular synthesis to stop, so we stop making macromolecules. The lysosome can begin to leak, leading to the destruction of organelles. Fusing of cells, syncytium. Host cell function. Interference, which protects the uninfected cells, and antigenic changes on the cell surface. Pathogenic properties of fungi. Fungal waste products may cause symptoms. So waste products may be toxic to human hosts. Chronic infections provoke an allergic response. Example of this would be chronic fungal infections in which we have mold growing in our homes. Proteases. Proteases are enzymes that modify the host cell membranes to allow attachment of the fungi, and we see this with Candida. Aflatoxin is found in peanut butter and is normally found grown on grains or other plants, and Aspergillus produces this. So pathogenic properties of algae. Paralytic shellfish poisoning. Normally we see these dinoflagellates present in very high concentrations in what we call a red tide. Do you see that red tide there? And they tell you not to eat or even swim in the water during those times. And just like how they came in, they have to go out. So pores of exit, respiratory tract through coughing and sneezing, gastrointestinal tract through feces and saliva, genital urinary tract through urine and vaginal secretions, the skin, the blood, arthropods that bite such as mosquitoes, needles, or syringes. So here, this ties it all together for you. The portals of entry, number of invading microbes and how they adhere. And then once they adhere, they have to be able to penetrate. And once they're able to penetrate, then they can cause what? Damage. And once they cause damage, then they continue to move. Here ends Chapter 8.